Hi everyone, welcome. Thanks for being here. My name is Luiz. I'll say a few words in a minute, but first, Leah from the SGB would like to say a few words as well on behalf of the Stan government body. So Leah, floor is yours.
Hi everyone. So I'm Leah Comment. I'm from the SGB, and I'm here to welcome you to StanConnect for biomedical applications. Really excited about our speaker list here. Really excited about the opportunity to connect virtually while we don't have in-person conferences. But yeah, thank you to all the speakers and thank you again to the organizers. So let's get started.
Thanks. So I'm gonna project my screen here. I'm gonna show you a PDF for the general plan, but first, well, yeah, so let's talk about the general plan. So we plan to have 6 talks today. Each will be 15 minutes, and then we'll give people 5 minutes to ask questions. But these talks have all been pre-recorded. So that means that the person giving the talk is actually the person in the past. So the person in the present will be there monitoring the chat for questions. So if you want to ask a question during the presentation, please feel free to do so. And the speaker should be able to see your questions and then answer them. Right, so we'll have 6 talks. We have a small break in the middle. And then by the end, we'll have some half an hour for networking, just to chat. It seems we don't have, a critical mass of a big number of people to break everyone in breakout rooms. So we're just gonna, stay on and talk a bit, and then people will have the opportunity to, ask more questions and then, just go off the rails and talk about other things in general. Right. So the idea for this this version of or this instance of StanConnect came when I saw that we already had a session for simulation-based calibration and also cognitive science. And I thought, okay, biostatistics cannot possibly stay, out of it. We cannot miss out on having our own StanCon because, over the years, I have been on the Stan forum, the discourse forum, we were quite active. And I've seen wonderful applications in all sorts of fields, from pharmacodynamics to survival analysis, and there are a plethora of very nice applications. So I thought, okay, so let's try and get everyone in, or at least 6 people to give talks on as diverse topics as we can, to try kind of showcase just how useful Stan can be for biostatistics, right? So I think someone has their mic on, so if you could please close it, that would be good. Right, so maybe a few words about myself would be in order. I am a lecturer in applied mathematics at the School of Applied Mathematics at the Getúlio Vargas Foundation in Rio. So FGV is a bit weird in that most of the research-heavy universities in Brazil are publicly funded, but FGV is private. It's a foundation, and it likes to think of itself as a think tank, which is a name I hate, but it gives people the right idea about what we do. And yeah, I mean, I hope this also kind of puts FGV out there in the map, so you can send your students here to work with us, and we can also send our students to work with you as well. So what else should I say? I think we're okay. I'm gonna switch to the first presentation, which will be by Mitzi Morris. So Mitzi is a researcher at Columbia University. And I think it's fair to say that if Stan is an awesome tool, it's thanks in part to her and her efforts, especially on the lightweight tools, right? So she's responsible for Command CMD StanPy, and I think she has had a hand on CMD StanR as well. She can tell us more about that later. But now I'm gonna play this video. She'll be talking about the Byom model, which is a conditional autoregressive model, very useful in epidemiology. So without further ado, I'll play it now. Let me know if there are any problems.
I'm Mitzi Morris. I'm a member of the Stan development team, and I'm here to talk about coding in Stan, the Byom2 model for disconnected graphs. The Byom2 model is a model that's widely used in epidemiology for disease mapping. And the use case is that you have data which is coming from some map. So you have a map, you have regions in the map, and all of your data is collected with information about what region that data was collected from. So what we want to do is we want to use the spatial structure of the map to explain what's going on in whatever our phenomena of interest is. And to do that, what we do is we take our aerial map and we look at which regions are neighbors. This is a binary relationship, and it imposes a graph structure on the set of aerial regions in our map. Most of the implementations for the Byom2 model, including the implementations in Stan, require a fully connected graph, so that from any node in your graph, you can get to any other node in your graph by some path, which means that on your map that there are no islands, there's no big disconnected divide running through part of your map such that one set of regions are inaccessible from another set of regions. And this is problematic because many maps do in fact have disconnected subcomponents or islands. And the analyst has to build bridges and tunnels where there aren't any. And this, for one thing, it's kind of messing up your data and your graph structure, although in a relatively minor way, but it's really very difficult to do, and it makes your data that much less reproducible. So we want to extend the Byom2 model in Stan to avoid that, so that if you've got a map, and somewhere on that map, you've got a set of connected regions where you want to use the spatial structure for those regions to explain what's going on, we can just you can just feed the whole map into your model, and all the islands are the islands, disconnected components get the appropriate amount of spatial smoothing, and everybody's happy. That is, in fact, not very easy to code. It's conceptually much simpler than it is when I'm talking about it, because coding it is what I do, and making things complicated is unfortunately a part of the game. Here's a map for you. Here's the United Kingdom. On the left is the map showing the regional boundaries, and everything on the left is colored by the component that it belongs to, or the island that it is. So you have two connected components and a couple of islands. And on the right, we have the map overlaid with the adjacency graph. So you can see that some regions have lots of neighbors, other regions have fewer neighbors, some regions are islands. And what you have to do in order to get all this information in the first place is that you're going to need to go to somebody's geographic information system and get the digital map, the map that has all of the coordinates, the geolocated coordinates that give you the lines and the points to make the shapes that are each each area in the middle. And then once you've got your shapefile, you're going to need to compute the adjacency matrix. Now there are packages that do this for you in R and Python. I was using the R package spdef, and that will definitely give you many ways to get the adjacency matrix out of your shapefile. Once you've got the adjacency matrix, you then need to take that and compute the components. So if you have disconnected components, you're going to get two components, and every island will be its own component. And then finally, you've got to figure out how to take your animal data, the information you're getting out of this shapefile, and make sure that everything lines up with however that regional information has been coded in your data. And finally, because in the STAN program, when we're representing this adjacency graph structure, all we have are vectors, and the entries in those vectors are just indexes into other vectors. So we don't really have any names that we're carrying around. We're relying on the implicit order of the way that the data comes in to inform us about which node is which. And this makes it very easy to introduce coding errors into your data where you get crazy results because the entry at index 5 really doesn't correspond to the region that you think it is. The Byom2 model is a successor to a long line of spatial models. And the Byom2 model takes a spatial component and decomposes it into the following. You have your spatial component phi, which is the iCar component for, I don't know why I said for fully connected aerial map. It's your iCar component, and your vector theta is ordinary random effects. So we want to explain on the regional, at the per region level, we want to explain both the spatial variance and the extra spatial variance for that region. So we compose them into a single component with a single amount of variance on the entire composed component. And then we add a mixing parameter row, which tells us how much of the spatial variance comes from our spatial effects and how much of our spatial, our per region spatial variance comes from, is not spatial actually. So again, sorry for the confusing presentation here. It's difficult to talk about this component coherently. And then what we need to do is we need to make sure that phi and theta are comparable. And that means that they have to be on the same scale. Now, theta is a vector of ordinary random effects, Gaussian 0, 1, 0. So the variance, the average variance of every element of theta is going to be 1 by design. All we need to do is we need to look at the spatial component, the iCar component, which is this sparse adjacency matrix, and we need to scale the adjacency matrix under line phi so that it too has a standard variance for each row in that adjacency matrix. The average variance is 1. And Riebler suggests doing this by taking the geometric mean of the adjacency matrix, and that's great. That's what we want to do. And then in 2017, Franny Starantino wrote this note on extending the Byom2 model to disconnected graphs. And her conclusion was that we want to scale each connected component just as we would scale a fully connected adjacency graph, adjacency matrix. And for each fully connected component, we want it to have a sum to 0 constraints because I failed to mention that you need to have a sum to zero constraint on your iCar component. Otherwise, you run into serious non-identifiability issues. And then for your islands in your map, you're just going to give them spatial variance that's standard normal 0, 1. So then you're going to have, again, like you have in theta, your vector of ordinary random effects. Now you can define as a number of iCar and random effects, and since each one of them is scaled accordingly, everybody on the left side of that equation of combination spatial, extra spatial, everybody in the spatial component is going to have an average variance of 1. And this is conceptually actually really simple, but there's some difficulties in it. So I'm going to present a Jupyter notebook that shows you what's going on for how it is currently coded in Stan. And this is perhaps not the best encoding because it's not quite as efficient as it can be, but it's my second attempt at coding this model. And we hope that the third time really gets it. So this is a Jupyter notebook. I'm going to put it in the GitHub repo. And it is using the canonical test data set for all of these Byom2 models, which is the Scotland data set of lip cancer in Scotland, 1960s and 1970s. And let's look at what we're doing for the fully connected Byom2 model in Stan. What we do in the fully connected Byom2 model is we can represent our vector phi and our vector theta as these standard as two vectors. And then when we want to combine them, I'm sorry, let's look at small ones. We've got the Riebler et al. Formula. Here we have to combine the square root of rho scaled by tau with phi and the square root of 1 minus rho times theta with the overall sigma variance component. What we do when we write the Stan model is we set up the parameters. In the transformed parameters, we create a new variable gamma that encapsulates this rather complicated formula. And then when we have the likelihood, we can clearly see what's going on. Then we have a function where we define a standard iCar component. And here we're computing with the adjacency graph, adjacency array, where we have a 2 by n structure. And I bet I'm running out of time because this is kind of complicated. Good. Let's keep going a little bit longer. We have these two adjacency arrays, and then we do this is the key to the iCar component. It's a pairwise difference model, and then we impose a soft sum to zero constraint on our iCar component, and we code this up as its own standard iCar prior, and then when we have the model, we just assign this prior to phi, and then everything runs and it's removed. For the disconnected graphs, suddenly things are getting a little bit more complicated because we need to keep track of the node and edge lists for each components, and this is the choice that I made currently. And the data looks really scary complicated. And the computation of the iCar component is similarly a little bit, not that much more complicated. But what we're doing is we're creating a vector of node indices, nodes. And then we use that when we are computing things with the, and we do the same thing for edges. We use all of this complicated indexing to create a list of edges. And so then when we can, then when we want to do the iCart component, which is right here, it looks pretty much the same, only we have to have some more, instead of just using the whole row 1 of the adjacency matrix minus row 2 of the adjacency matrix. Now we have this component, this edges, which is picking out individual elements. And that is basically where we're at right now. Thank you very much. And let's discuss more in the question sessions. Thank you again, and looking forward to your questions.
Right. So that was Mitzi's presentation. Thank you very much, Mitzi, for accepting my invitation and also giving this presentation. If you would like to say a few words more about the implementation, and of course, if anyone has questions, please do ask them now. I forgot to mention a small thing, which is we got Frank Harrell to discuss the presentations. He will be, he'll have some time in the end to say a general sort of address, but, I'm sure he'll have questions now as well. So, Mitzi.
Okay, so thank you. And I hope this was what you were expecting, but what I was trying to get at in this presentation, the punchline, which I can give to you in person, is that when you're doing the kind of indexing that's currently being done in this model, it's less efficient because you have to pick out individual elements. What makes a Stan program fly is always vectorization. So what we really want to do is we want to find a way to have things line up so that you can cleanly slice your vector. And then you've got a bunch of stuff that's all adjacent, and that gives you really, really great properties when it comes to computation. So this talk is, I was trying to get at the difficulties that I've had in putting together what should be something very simple and making it usable, because the amount of data munching on the outside and the amount of computation on the inside is just rather daunting. And my goal is to always create general purpose usable tools that other people can use. So that's where things really get difficult. If this was just my data and my analysis, no problem. But how can we generalize that for everyone?
Right. I had a statistical question. I don't think I think I may have missed something. In the disconnected model, what constraints do you have on the disconnected components? Do they, does there have to be some global scaling, or can you kind of treat each disconnected thing?
Oh, right. Okay, so if you remember in the Byom2 model, there's that scaling factor tau.
Yes.
So in the disconnected components, for each component, there's a tau associated with that component.
Right.
So then everything scales, and this was a very challenging. The other challenge that I've had in putting together this model is that the paper that describes it, the Freni-Starantino, is not quite, it's written by a non-native speaker writing in math. And so there's a lot of problems I've been having interpreting the math and interpreting what she says, because there's recommendations, and the recommendations are clearly not quite fluent. And that's really a problem when you're trying to express a mathematical solution. What exactly do all of these quantifiers mean? My background is linguistics. So, deciding what amount of scaling you need has been for me a big challenge. but I think I've sort of powered through it, and it's really much simpler than I made it the first time around.
Right.
Scale each component accordingly and make everything sum to zero, and it really just should all work.
Right. So thanks. Teddy has a question. Would it be easier to code this model if Stan had a sparse matrix data type?
Actually,
I've seen that thrown around a few times. Go on. Sorry.
what would make this particular model, as it's currently implemented, easier to code would be if Stan had ragged arrays.
Right.
If Stan had ragged arrays, then I wouldn't have to do the work that I was doing with some of that indexing. But even if we had ragged arrays, that's not really what we want to do here. So the answer is sparse matrix types are not necessary at all for this model because you've already computed the sparsity on the outside. The driving feature of these kinds of spatial models is that as the number of regions in your map gets larger and larger, things get more and more sparse. And that's why all of the SP-DEP representations and all of the representations of your neighborhood network are already various kinds of sparse encodings.
Right.
Yeah, and going off of what you said, Mitzi, and actually I see that he's in the audience now. Sean and I were working on this GLM for copulas thing, and it's possible that like if you have say 3 outcomes, that the design matrix for each outcome is different. So that's fine, all is well when you're coding specifically for 3 outcomes. But if you wanted to generalize that code, say to capital J outcomes, and each one could have a different design matrix, it's like very hectic to actually implement that in Stan. And also, we ran into another problem where doing this copula approach requires CDF evaluation at possibly non-integers, and you can't even do that in Stan, nor is that CDF like vectorized. So I don't know, just thoughts for room for improvement.
Yeah, I bet. Yeah.
Well, just speaking CDF, vectorized CDFs may come sooner than later, but
yeah. I mean, it's interesting. Stan is such a mature project, but there's still a lot of stuff that we still have to do, to get our models to work. I'm keen to see some phylogenetic stuff as well, but not by me because it's really hard to code. Right. So does anyone have any more questions for Mitzi? Otherwise, we can move on to Ethan's presentation. I'm going to try something slightly different. I'm going to share the video specifically. I'm going to play it, and I'm going to mute myself, and we'll see if that works, right? So you'll let me know if that works well. And if it doesn't, we'll switch back to the old thing that works for Mitzi's presentation. Right, so Ethan is a recent graduate from UNC, recent PhD graduate. He's an incoming researcher at Byom2 Women's Hospital, Harvard University. He'll tell us more about that later, but now I'm going to share his presentation on the normalized power prior, which I'm a bit biased towards because I'm a co-author in there. Right, so that is Ethan's optimize.
Hello, everyone. My name is Ethan Alt, and I'm an incoming investigator at Byom2 Women's Hospital and Harvard Med.
Does that work?
I think everyone needs to mute themselves.
No, it's probably me. Let's see.
And recent PhD graduate from the University of North Carolina at Chapel Hill in biostatistics. And today I'm going to be talking about normalized power priors in Stan. So to provide a brief overview of the things I'm going to talk about today, I'm first going to introduce the power prior and the normalized power prior. I'll then discuss a particular implementation of the normalized power prior in Stan that I have been working on with some colleagues. I will briefly present some results and examples and then provide a conclusion. So to introduce the power prior, which was developed by Ibrahim and Chen back in 2000, is a prior based on the likelihood of historical data. So if we let D0 represent historical data and suppose that the data generating process is parameterized by a vector theta and a likelihood function, the power prior is really just given by a discounted likelihood, which is this likelihood function here, and an initial prior pi0. And ZA0 is simply a normalizing constant. So we typically take A0 between 0 and 1 so that the prior doesn't have more influence on the posterior than the current data. The hyperparameter A0 controls for the level of informativeness or borrowing of the priors. For example, if A0 is equal to 0, then the only part of the prior that enters is the initial prior. If A0 equals 1, then the prior is the posterior density of the historical data. So the key question is, how do I select A0? Well, one thing that you can do is simply try several values of A0 in some sort of grid, say A0 equals 0.1, 0.2, and so forth. But from a regulatory perspective, this isn't a viable option because regulators are going to force you to choose a specific prior and not let you pick and choose whichever value of the hyperparameter fits the best. Another approach is to treat A0 independently as random by specifying a hyperprior, which is just a distribution over the parameter space of the hyperparameter. So for example, we could specify a beta prior for A0 or any other prior that has support between 0 and 1. And this yields the joint power prior, which is given as this discounted likelihood, the initial prior, and a prior over A0. The problem with this approach is that posterior propriety is not guaranteed, and one can show that such a prior disobeys the likelihood principle. So the solution is to use the normalized power prior, where in fact we have to include this normalizing constant that depends on A0 inside the prior. Now, the reason why this is somewhat complicated is that this normalizing constant is not known in general. And for regression models, the only one in which it has a closed form is the normal linear model. For any other even generalized linear model, Z of A0 is unknown, and therefore we have to develop a method to estimate Z of A0. So the normalized power prior, once again, is simply given by the discounted likelihood multiplied by an initial prior that depends on the parameters for the model, multiplied by a prior for A0, which is the power per prior parameter, and we have a normalizing constant, which is defined here. As I previously mentioned, for regression settings, the only case where Z of A0 has a known closed form is the normal linear model. So the key question that we're going to try to answer in this talk is, how can I obtain samples from the posterior if we use pi NPP, that is the normalized power prior? So we offer two solutions. One is more general than the other, but one is much quicker than the more general solution. We'll first talk about a pure Markov chain Monte Carlo approach. And again, recall that the parameter space of A0 is between 0 and 1. We can define a small grid of values between 0 and 1, say 0, 0.05, 0.10, and so forth. Then for each of these values in the defined grid, we can sample from the conditional power prior, that is treating A0 as fixed. So for example, if A0 is 0.05, I'm just going to sample from this prior distribution, which is a very easy thing to do in Stan. When I do that, I might obtain M samples, say 2,000 or 10,000 samples. So then we can use bridge sampling via the bridge sampling package in R to actually obtain an estimate of the normalizing constant at this fixed value A0. Another approach is to use importance sampling. We know that if the sample size is large, that the prior density really is going to converge to a normal distribution, where the mean of the normal distribution is the posterior mode and the covariance matrix is the negative Hessian. So the point is, this approximation is going to work well when all the parameters theta are approximately bell-shaped, symmetric, and unimodal. Stan actually offers a built-in optimization routine. So once we program the power prior, we can use the optimization methods in Stan for free to actually find theta hat and sigma hat. Once we have theta hat and sigma hat, we can simply turn to importance sampling to find an estimate of z of a0 for any fixed value of a0 in the following way. So all I'm really doing here is I'm drawing a theta hat from this normal distribution, and I'm estimating the normalizing constant via this importance ratio, where phi is the multivariate normal PDF. So long story short, this gives me a good estimate for the normalizing constant at a fixed value, provided all the components in theta are approximately bell-shaped and unimodal. So in terms of how can we actually implement this in Stan, the first thing to do is to define a grid, A0, and use either the full MCMC approach using bridge sampling or the importance sampling approach to obtain estimates for the normalizing constant for each value of the power prior parameter. Then, because of Monte Carlo error, that function may be relatively not smooth, and in addition, it's not evaluated at so many points. So you could either use a linear model with polynomial terms or some other fancier method to obtain a smooth estimate for this normalizing constant for a very fine grid, which I'm going to call A1. So now we have basically a data set or a matrix that gives you a value A0 and an estimated normalizing constant z of A0 for every A0 in this set A1. That's the prior stage. Now, in the posterior stage, we're going to have to create a function in Stan that conducts linear interpolation based on this grid of values and the estimated normalizing constants that we use. So we're basically just going to define this function in Stan that will take in a normalizing, a power prior parameter and output a linearly interpolated value for the normalizing constant based on the input. And the interpolation is going to be based on both the fine grid defined in step 2 in the prior stage, as well as its estimated value from the smooth curve. So once we have a way to estimate this normalizing constant, then everything in Stan is quite easy to do. We simply increase the target density by likelihood of the current data, the discounted likelihood of the historical data, the initial prior, the prior density for A0, and finally the estimated normalizing constant. So here is an example where bridge sampling and importance sampling essentially produce the same results for the normalizing constant. And this is likely due to the fact that the sample size is large relative to the number of parameters, in addition to the fact that all of the parameters in the posterior are symmetric and unimodal. And here is actually a plot of the parameters using both the bridge sampling method and the importance sampling method. And what we really see is that the posterior density does not differ much between the two methods. So in this case, the importance sampling method, which is much faster than the bridge sampling method, essentially produces identical results. But this is, of course, not always the case. When there are many parameters relative to the sample size, or even if there are some parameters whose posterior density is not symmetric or unimodal, say a dispersion parameter, then the bridge sampling and importance sampling results will be quite different. And as we can see, the bridge sampling produced a mostly symmetric and unimodal posterior, whereas the importance sampling is neither symmetric nor unimodal. And that would suggest that the normalizing constant was not estimated that well. So in such circumstances, we would prefer the bridge sampling method to estimate the normalizing constant. And finally, there are cases where bridge sampling and importance sampling do produce similar results, but neither of them are quite good enough to actually be utilized. So for example, here it seems that the log normalizing constant kind of behaves erratically as A naught changes. And indeed, when you look at the posterior density of the regression coefficients, you see that they're neither symmetric nor unimodal. So to conclude, we have offered implementation of normalized power priors for all generalized linear models. Problems can occur with importance sampling when there are dispersion parameters, and this results from the fact that many dispersion parameters, especially for low values of A0, are not going to be symmetric or unimodal. In general, highly parameterized models will likely not work well, and for such models, I think we will just need to take an extraordinarily large number of samples to really estimate that normalizing constant well. In addition, perhaps a finer grid. It is possible to implement a Laplace approximation inside the sampler to avoid a two-step process, and that's something that we are currently exploring. And finally, we are considering possible extensions to survival data. And here's the one reference that I had. Thank you very much for listening.
Right. Thanks, Ethan. So if anyone has questions for him or for me, because I'm a co-author in this as well, we would be happy to answer. How did that go, though? Did it go all right? Did you listen? Was the sound weird? Okay.
It sounded weird to me, but that's just because it's my own voice. So it was very clear.
Great. Okay, so we have a question from Sean. So Sean says, are multimodal posteriors just sometimes inherent in the model and you would never get good estimates for the power prior? Yeah, that's a
good question. For the vast majority of cases for generalized linear models, especially for regression parameters, it's highly unlikely that the posterior should be multimodal. And I suspect, Luis is actually the one that ran that scenario. And I think what it basically was, there was like 50 or so regression coefficients. Yeah. So the model was just like really highly parameterized. And I think when in such a case, one just needs to take perhaps a prohibitively large amount of samples in order to actually get a good estimate of the normalizing constant.
Yeah. So it's surprisingly easy to break marginal likelihood estimators, right? One thing you can do is just increase the dimension, which is what I did here, right? So we're dealing with simulated data in a Poisson GLM, and I just made the design matrix instead of 5 predictors, I just used 50, and then everything went to hell. But the good news is that particular problem can be fixed relatively easily because, if everything stays the same, then you can use the sort of sparsity and the structure in the design matrix to sort of get better estimates of the marginal likelihood. But the problem is that you might have a model with 50 predictors, right? And with very complicated relationships. So you're not going to be able to fix that. The upshot is that sometimes neither method or no method we know works. So that's kind of an open problem anyway.
Yeah, I think with any of these types of models where the normalizing constant needs to be estimated, I suppose that really a better tool would probably be old-fashioned MCMC, where one can come up with tricks to make the normalizing constant not be dependent in the sampling scheme. It's not really possible in Stan, as far as I know. They're called like so-called doubly intractable posterior densities.
Yeah.
And as far as I know, there's no Hamiltonian Monte Carlo way that's been developed to actually handle such things implicitly in the way that like a Metropolis sampler could.
Yeah, there's this thing called noisy HMC that kind of gets at that. But the problem with all of these algorithms for doubly intractable problems is that every single one of them has a catch, right? So for instance, the exchange algorithm works very well, but you have to be able to generate samples from the likelihood, which in our case wouldn't be possible. So yeah, it's a hard problem, period, right? All we can do is fix the problem in small self-contained examples, and I hope we can do that for GLMs. So we have one last question by Martin. So Martin asks, what are the main use cases for, I guess, normalized power priors? I'm going to let Ethan answer that, because his supervisor, his PhD supervisor, was the creator of power priors. So I think Joe would like you to.
I said putting my fresh PhD to use here. Well, the main use cases and the reason why this was developed is mostly for clinical trials, where you have historical data and you would like to use that historical data to inform, say, a treatment effect. In terms of why not just do a normal prior based on the MLEs or something like that? Well, you can do that, but the key advantage of the power prior is that you can discount the likelihood, so that way it won't depend on the historical data too much. And then you might ask me, okay, well, why don't I just keep A naught fixed, or why don't I just use what's called a asymptotic power prior, where they will use the MLE and the covariance matrix to construct a multivariate normal prior on the regression coefficients. You can do that as well, but of course, the asymptotic power prior is just that. It's an asymptotic result. And kind of like the key advantage of the power prior is that you are able to elicit a prior that's correlated, whose components are correlated a priori, which is nice, say, if you're interested in effect heterogeneity. And you can control the level of informativeness. And the reason for treating A naught as random is it kind of gives you an idea of how compatible your future and historical data sets are overall. So that way, if your data sets aren't compatible, one would hope that A0 would be selected close to B0, so that way you actually don't end up using that historical data.
Right. So thanks, Ethan. There are a couple more questions in the chat, but I don't want to take too much time with so if Ethan, if you could step into the chat and help answer some of these questions, I can help as well.
Yeah.
Sure. I'm gonna now share the presentation. So give me a minute. So we have two people from the Technical University of Denmark. Teddy Groves is a postdoctoral researcher, and he'll be the first. His presentation will be the first from this series. So let me just find my footing here. Good. Okay. I'm gonna now share this. We're steady. All right. So you can see that, but it wasn't up to, okay, cool. Done.
Hi, my name's Teddy Groves, and I'm a postdoc in the DTU Center for Biosustainability. I'm going to present some work I've done with my colleague Areti Sigkinopoulou about analyzing information from online databases about enzymes using STAN. So to start by a bit of background, since about 1920, it's been possible to mathematically describe how enzymes work. So, and this is really good news because enzymes are really useful for lots of different things. So, yeah, if this is an equation which says in terms of some physically interpretable numbers, like the concentrations of some chemicals, enzyme concentrations, how fast the reaction on the right goes, and so it's really great that it's possible to parameterize enzyme catalyzed reactions in this way because this makes it possible to predict how an enzyme will behave in a new condition, which is really useful for if you want to do things with enzymes. But in order to make those predictions quantitative, you need to find what the numbers are. And the concentrations you can measure pretty easily, but the numbers beginning with K are called kinetic parameters and are a bit more tricky. But they can be measured, and lots of measurements are available online from databases like Brenda. So this means it's basically possible to do quantitative modeling of networks of enzyme catalyzed reactions. But even though it's possible, it's not necessarily easy because the measurements that you find in online databases often disagree with each other. Then it's often hard to access particular bits of information that you want. So it's quite common to have to read the actual paper whose results are reported in an online database so that you can find out things like the experimental conditions or the exact nature of the enzyme that was tested. Then also often kinetic modelers have to rely on sort of inference by analogy. So even if a particular kinetic parameter hasn't been measured, maybe a kinetic parameter has been measured that is similar in some way. And often the modelers find themselves using, for example, a kinetic parameter from a different but similar organism in the same reaction. Yeah, so basically the upshot of this is that there's a sort of step 0 before you can start doing any kinetic modeling, which is very labor intensive, involving going through databases and sort of making lots of judgment calls. So we thought that it might make this process a little easier if there was a statistical model that would summarize a lot of the information that's available in the online database. So we thought we'd make one for KM parameters and the Brenda database, and then make the results easier to access than the ad hoc process that I described previously. So what we did was first fetch some data using Brenda's API, then process it, and we did some quite aggressive data processing, like only keeping one observation per kinetic parameter per study. That was because different studies report different numbers of measurements in Brenda, and it doesn't necessarily correspond with how much work they did. So often a study would have done like loads of work to find out one kinetic parameter, but only report one number. Other times a study would have done like 20 measurements of the same kinetic parameter, but under slightly different conditions and with slightly different numbers. Yeah, so we thought the best thing would be to keep one per study. It's still not perfect, but you know, we thought it was the best option. Then to make it easier to iterate through models, we removed some data by only keeping measurements from human yeast and E. coli, which still leaves us with a large data set, but more manageable. And we only kept measurements of enzyme substrate combinations that occur in nature, because those are the ones that are most interesting to kinetic modelers. And what we ended up with was the graph on the screen, where the log kms go from like minus 17 or 18 up to like plus about 6. Yes, then we had to pick a model, and we spent a long time doing this and went through loads of models. And then the one that did the best was this one from this paper by Borger, Liebermeister, and Klipp, which says that the best predictor for the log scale Km value is a global mean plus a term for the substrate plus 2 interaction terms, one for the interaction of the organism and the substrate, and one for the interaction of the enzyme and the substrate. So we wrote that model in Stan as a hierarchical model with you know, hierarchical priors on those alpha parameters, and otherwise with fairly normal independent parameters, and the student T measurement model. Yeah, and this is what happened when we fit the model to some fake data. It worked okay. Yeah, this data was generated using the same Brenda data set, just with the measurements generated using the model assumptions. Then this is the results when we fit the model to real data. You can see that the posterior predictive distributions are smaller for the posterior than the prior, which is good news, because it means that there was some information in the data that we saw. Then to see how the model would perform with out-of-sample predictions, we firstly tried doing approximate cross-validation using the Pareto smooth importance sampling algorithm, via which RViz provides a really easy interface to. But unfortunately, the algorithm raised some warnings. So we thought, even though we used these results a little bit for comparing different model runs against each other when we didn't have time to do full cross-validation, we actually thought that we should do some exact cross-validation as well. And yeah, this is the results of our exact cross-validation code. And yeah, the good news is that our final model did better than some very simple dummy models. So we think it's better than nothing. Yeah, so then to make it easy to use the results of our model, we made a web app. It's not deployed yet, but you can run it on your own computer by going to the project GitHub repository, which will be linked to at the end of these slides. And yeah, on there you can enter the organism, enzyme, and substrate that you're interested in, and see the marginal posterior distribution for the corresponding KM. And you can also download CSV with all of the model results in it. We also hope to address the issue of when a measurement is not available by adding some functionality to let you put unknown, yeah, make predictions based on what you do know. So that's what we did. It's still a work in progress because we haven't deployed the model yet. Oh, sorry, deployed the web app yet, and you know, or even really finished the paper. But we're quite happy with how it went, even though basically the data is pretty tricky to model. Yeah, there's definitely room to make a better model of this data, and any suggestions would be really gratefully received. I thought it'd be good to point out how nice the file-based workflow with command StanPy is. So in this case, we had lots of different ways that we wanted to use the same model. So the prior mode, the posterior mode, the fake data mode, and the exact cross-validation mode. And having files makes it easier to keep track of that. Or at least it did. I thought it did. Then one problem that we had was that because our model has a pointwise log likelihood and the posterior predictive, a variable for each observation in the posterior predictive distribution, it means it has loads of parameters and the I/O takes a bit long. So yeah, anything that would speed up the I/O would make our life a lot easier. And then finally, we made the project initially using a cookie cutter template that I wrote for command StanPy projects, and the experience of doing this basically convinced me that I didn't write a good enough cookie cutter template yet, because we had to make loads and loads of changes, and the final result looks very little like the template. I still think it's fundamentally an okay idea, but it takes quite some thought to make a good template for command StanPy projects, I think. So yeah, thanks for listening. Please check out the GitHub repository and feel free to contact me or Areti on these email addresses. And yeah, lastly, I should say thanks to the Novo Nordisk Foundation, who provide the funding for our research group. Yeah, and thanks again for this.
Right. So that was Daddy's talk. Daddy, if you want to say a few more words, if people have questions, we have plenty of time for those. Go on.
So some people said, what is cookie cutter command StanPy? So cookie cutter is a Python library that is meant for writing project templates. And I had a thought a while ago that it'd be really nice, rather than writing a lot of the same code over and over again, to make a template for a command StanPy project that would kind of put you in roughly the right place when you're about to start a new project. And generally, I think project templates are a really great idea, because basically it's a lot less work to maintain a template than it is to remember all of the things that you should remember every time that you're starting a project, like the license and the all of these like TOML files that you should put in the Python project, and all of these things that actually make your life a lot easier. But if you have to think of them every time you're starting a new project, it takes ages. Whereas I know there's some cookie cutter templates out there, like I don't know, like cookie cutter data science or something, that are quite widely used and generally thought of as a good idea for when you want to start a new project. So I had a go at making one for Commands NumPy, and I think I posted it on the Stan Discourse. And Mitzi said, I think it's a good, good general principle, but since everyone's project is slightly different, it's really hard to make it work. And experience has totally borne that out, because even though I wrote the template myself, when I actually wanted to use it, I had to deviate from it quite a lot. So there's some half medium to be found, but I guess it has to just be like pretty minimal, the cookie cutter.
Yeah.
But I think it's an interesting topic generally.
I mean, if it can be shared widely, publicly, people can then contribute to it, and maybe you can have some different flavors. I don't use Python a lot. I use MESSIR, which is its own special language, right? It's just a bunch of scripts that I cobbled together from other scripts in other projects. It's amazing for bugs, man. It does a great job at introducing bugs that are untrackable, pretty much. I had a statistical sort of more scientific question, which is, you say you did some pretty aggressive data trimming, right? So data processing. What's the risk of missing some important information there? So for instance, if you have a study and there are multiple measurements and you want to sort of have an idea of what the intra-study sort of variability is, if you only keep 1 point per study, does that kind of hamper, are you missing some information possibly in there?
Yeah, absolutely. I don't think it's a choice between bad options, basically, because the data is bad. So it's not necessarily the case that one number in the data corresponds to one scientific measurement.
Okay.
It's not possible to recover the original measurements.
Right.
So we're faced with the choice of, I think maybe we could have done something sophisticated where we try and like take into account the study. But yeah, we didn't get anything like that to work.
Right.
But you're absolutely right. It's there's going to be bias, in there one way or the other.
Right. Yeah. I mean, it's I think the important bit is to try and get a hold of just how biased and in what direction, right? Because maybe it's going in the direction of, I don't know, higher quality studies, and that, I don't know. But there's some confusion there that would be nice to account for. But so the other question I had was, how do other people actually use this? What are the uses that you envision for that, nice application of yours, where you visualize the marginal posterior for the quantity of interest?
Well, Nick's talk is going to actually go into this a little bit, because one of the main uses for this kind of analysis is to get priors for a statistical model of a metabolic network.
Cool.
Because the kinetic parameters are a really important component in that kind of model.
Nice. Have you considered power priors? I'm joking.
No, actually, it's the first time I've heard of power priors. I need to check out that paper.
Yeah. Okay. Does anyone have any more questions or comments about this particular talk? Well, if not, then we can have ourselves a break. Since we're doing well for time, we could do with some 10 minutes to, grab a bite to eat. Well, it's lunchtime here, so I'll definitely try and run to the kitchen and see if I can find something there to scavenge. But then we'll come back and we'll have 3 more talks, and then Frank will say a few words if he wishes, and then we'll have free time to just chat, talk to each other, ask more questions. So if that sounds like a plan, I'll see you in exactly 10 minutes. So that will be 11:22 Eastern, right? Okay. See you soon. restart this recording thing. And we should now be ready for a second round of talks. We will now have a talk by Teddy's colleague, Nicholas. So Nicholas Cow is a PhD student at the Technical University of Denmark. And I will now project his talk. Let me give me just a second. So go. Okay.
Good morning or afternoon, everyone. I'm Nicholas Cowan, and today I'm going to be presenting the automated kinetic modeling in STAN and its applications to the Methionine cycle. So if you're familiar with any biochemistry, which I'm not assuming you all are, this is one of the most simple kinetic formulas there is. It's called the Michaelis-Mentioned Equation. And what it describes is the binding of a substrate to its enzyme to form this enzyme substrate complex, and then the catalysis of this to its, and dissociation to its product and recycling of the enzyme. It was kind of constructed by Michaelis and Maud, both scientists. This is Maud Menton. She was the co-author of this, and she's the first doctor in Canada, and she moved over to Germany to kind of work on these enzyme kinetics. So just to visualize the behavior a little bit further, this KM represents this half binding rate where at 50% of the rate, you'll have this amount of concentration. And one other thing to note is the rate is asymptotic with concentration. So as you take it towards infinity, you only begin to approach this Vmax value. So of course, enzyme kinetics can get far more complicated. You have multiple substrates interacting. You can have reversible behavior. However, for the kinetic modeling aspect of our software, we decided to standardize it to primarily the modular rate law. And it's very similar to the Michaelis-Mentioned kinetics, and we can account for additional things such as allosteric regulation, phosphorylation, and if you're very desperate for a specific law, then you can include things like kind of better representations for your own model. And our reasoning behind using a standardized rate law for the software side was there's a lot of research burden as well. There are many different laws out there, and a lot of them require a lot of research to try and understand. So for each enzyme, you can have a different binding mechanism. And with this standardized law, you can achieve like roughly like 70% correlations compared to more complicated ones. And in biology, that's generally good enough. So with these representations of kind of enzyme kinetics, we can connect them together and try and describe how our metabolites are. So metabolites are the kind of substrates and products within a metabolic pathway, and the fluxes are the fluxes between these. And when we look at a metabolic pathway, we assume it's in the pseudo-steady state. So in reality, a lot of these metabolites influence the transcriptome, which then influences the protein concentrations, and the proteins are our enzymes, which catalyze these reactions. However, when we take this snapshot, the protein concentration doesn't change, even though metabolite concentrations are kind of changing over time and reaching their steady state. And we can define a growth condition and a protein concentration and get out our fluxes and metabolite concentrations for a steady state. To kind of visualize this and see it clearer, this is the methionine cycle. It's very important in human metabolism, and its metabolites are used a lot for kind of different building blocks of your cells. So a lot of nucleotide synthesis or certain redox pathways. And because it's so central to a lot of cellular behavior, it's very highly regulated. So a lot of these metabolites need to be at a specific concentration for a cell to operate properly, just to maintain, for example, a thermodynamic driving force down a pathway, or if these metabolites are effectors of other pathways, you don't want to ruin some downstream pathway just because you need to increase nucleotide synthesis, for example. So this phenomenon in biology is known as homeostasis, where you can have a lot of different fluxes for very similar metabolite concentrations. So the bold represents the enzymes, which can be isoenzymes. In this instance, we have two enzymes catalyzing the same reaction, or you can have two enzymes catalyzing almost similar reactions with different cofactors. And the normal font represents the metabolites. The dotted lines are allosteric interactions, and they're quite prevalent in this pathway. And yeah, so this is a methionine cycle, which we then made into this set of ordinary differential equations, where we define our metabolite concentrations as the sums of all the different fluxes into and out of each metabolite pool. And we solve this system as an initial value problem. So in an ideal world, we would construct our kinetic model, we would measure our kinetic parameters and have priors for them, and then predict cellular behavior. One of the things why we don't have very strict priors is these measuring the kinetic parameters. It's very hard to do properly because the typical way you would measure them is in vitro, so out of the cell. And enzymes have different characteristics when they're inside the cell versus when they're outside. So, for example, you would have specific ion concentrations, or they tend to bind in a certain way, which you don't typically observe when you take them out of the cell. So, unfortunately, we can't do this. So this is a simulation of the methionine cycle. These are the metabolite concentrations, fluxes, and enzyme concentrations. And the light blue bars represent the marginal distributions. The marginal distributions using only our priors on the kinetic parameters. And the black ones represent the marginals of the prior given the measurement as well, the joint posterior distribution. So one thing to note is using only the priors, you have very dispersed metabolite concentrations and fluxes. These are on the log scale, and some of the metabolite concentrations go over over 20 orders of magnitude, which is quite ridiculous. And a lot of these fluxes go between like 10 and 0, which is quite a large range compared to the posterior distribution, which is very close to the measurements. So you would expect that given this shrinkage in kind of posterior compared to the prior distributions, you would expect a lot of constriction on the kinetic parameters. And we don't actually observe this. So there's a few kind of reductions in the marginal distributions on the K-cats and on the K-Qs, but generally there's not too much change compared to the prior distributions. And we believe that has several kind of causes, which we'll get to later. So this is just a rough representation of the statistical model that we make. So we have all of these which represent our boundary conditions and kinetic parameters, and we can get priors for these from experiments or from literature. You can also input things such as the stoichiometry, allosteric interactions, and phosphorylations, which go into determining this system of ODEs. And then given our boundary conditions and parameters, we solve the initial value problem to get our measurement, which we then compare to our predicted value, which we then compare to the measurement from our experiments. One thing to note is that this transformation from the boundary conditions to the predicted values is nonlinear. And we would like to put priors on some of these values. However, unfortunately, this is, if anyone has any ideas about that, that would be lovely. Our software called Maud takes the following inputs. So we have priors defined as a CSV, measurements as a CSV, and sample instructions and kinetic model definition are defined in TOML. We went with TOML just because of how readable it is, and it's just a nicer thing to look at than JSON or SBML. Humans interacting with SBML just should not be a thing. And then this is all passed through into Stan, which automatically constructs the ODE system for the kinetic model, given the user input, and then performs a HMC sampling and also does the kind of generated quantity section, which can be anything from simulating new experiments or simulating measurements from this experiment that we just did. And then this is passed to the output, which is a directory containing all of our samples in a CSV and RViz inference object, any debugging information such as did everything reach its steady state, and the input directory so the user can always replicate the same sampling as they did previously. And our kind of workflow would be, these are the different experiments that you did. You pass it to Maud, you generate your kinetic sample, your posterior samples, and then you can use this for anything from strain design, regulatory contribution, or are you missing any regulatory interactions? And yeah, this is generating these statistically sound parameter sets, which is, I believe, very central to it. And some of the challenges that we face in our automated construction are these high dimensional correlations. They result in difficult posteriors. So this is for a single enzyme parameter set. So the kms for every metabolite within that enzyme, and kcats and equilibrium constants. And what you can see is they have these very kind of almost hyperbolic like structure sometimes, or in this instance, you have the like almost a boundary that gets more dispersed as you kind of move away from these lines. I believe there's kind of even higher order interactions, but it's very difficult to visualize, as most of you know. So this makes the sampling very difficult. Often you'll encounter divergences in your samples, and there's a lot of challenge in actually iteratively building your models. So as many of you know, modeling is an iterative process. You have to go through it a lot of times to go, okay, is what I'm doing actually correct? Have any of these measurements, like, are they unable to be made? And how can I make my model just better as you go on? And kinetic models are difficult to evaluate as well, because you have to simulate the ODE for each step because they're evaluated, and they're made worse by this high dimensional correlations. And as far as I know, they're only parallelizable on the experimental level. So if any of you have any ideas about that, that would also be greatly appreciated. Thank you very much. I'd like to acknowledge my supervisors, Lars Nielsen, Teddy Groves, and Pedro Saar, and the rest of the quantitative modeling of cell metabolism group, and also the Novo Nordisk Foundation. Thank you very much, and I'm excited for the following question.
Right. So that was Nick. Thank you, Nick, for your talk. Does anyone have any questions for Nick? I certainly do have a few. Sorry? So Ethan asks, could you pre-process the inputs to reduce the correlations?
Sorry, I'm not too sure how much pre-processing of the inputs we could do. I mean, we normalize them so that they're going to be roughly around, so they're log normally distributed parameters, just so we keep them all in the positive space. And then we normalize them around the prior mean as well. But I'm not too sure how much pre-processing we could do to reduce those correlations.
Could you do like principal, I mean, if you don't care about interpretation of the inputs, could you do like principal components analysis on the inputs so that way they're like not correlated when you put them into the model?
Does that correlation, does it only remove correlations that are linear in their behavior?
Yeah, yeah, it does.
Because yeah, from what I saw, it's these dependencies, and I believe they're just degeneracies, like multiplicative correlations and things like this that we typically don't want to remove, because in some circumstances they're like you can't really observe them just because the experiment doesn't explore that area. But in other instances, like you change your experimental conditions and you end up exploring that zone a bit further. So it'd be nice to keep them all, but in terms of reducing them through PCA, I don't think it will be possible too much.
Right. Yeah, those boomerangs are nasty, right? And non-linear. Right, so
Yeah.
you see in the boomerang exactly where the derivative explodes, right? So that's why you get a bunch of divergences. Nasty problem. I'm gonna post a link to a paper that might give you some ideas on the prior thing, right? So I've dealt with this problem before. I'm sure some people here have dealt with that problem as well, which is you have a model, you have some inputs that you have priors for, and that model has some outputs that you also have some information about. And you would like to sort of, understand how to coherize, let's say, those two sources of information. That's what I gathered from your application, right? Which is, let me get their names here. C balanced and V reaction are quantities that get induced priors by the model, but also you have some information, right? That's the situation. So yeah, what you can do is something that looks a bit like the power prior, actually, where you raise both densities to a certain power, and then you take a sort of geometric mean between the densities, but it doesn't need to be, 1/2. You can choose the weight appropriately. So if you're interested in that, I've done some more work and I can definitely help trying to get this implemented.
Yeah, that sounds lovely. Thank you very much.
Right. More questions? Comments? Yeah, I was going to ask a general question, but I think you answered that in the course of your talk, which is how exactly do those hierarchical components interact, with the data? And you said you observe a bunch of divergences, right? And they don't seem to go away when you increase the step size or other things, right?
The divergences do eventually end up going away. It was primarily in just the model iteration thing. It'll be, I don't know, 5 divergences in like 500 sample chains. And you don't notice those until it's already done. And if that's taking a full day to do, and you're like, oh, damn, I thought it would work this time, then you need to start again. So that's the pain in there.
So we really got to find a way of, discarding things with divergences and, keeping the good work that you've done and not throwing it all away, because it's a terrible waste, right? So when you do hours of computation and you observe some divergences and then so technically I know I have learned nothing, but it doesn't feel that way, right? I should be able to reuse this information somehow. I know of certain efforts in that direction, adaptive path sampling and importance sampling to try to get just salvage what you can from the posterior and sort of get away from the divergences, but that's still quite early.
Yeah, I think our main objective would be to know if it was going to fail earlier rather than later. But I guess many people have that problem as well.
So Mitzi has a question. She says delayed rejection sampling, but I didn't quite get what the question is. Are you saying that delayed?
Oh, it was just in terms of how do you deal with the divergences?
All right.
And I think the question, the hope is that you can just get there's been everybody on the Stan team is very excited about this paper by Liu Zhang on Pathfinder, which is hopefully going to get you to the right start faster. But then if you have a very difficult geometry, you have a difficult geometry. Yeah,
there's not much you can do. And the problem is that, the Stan community general advice is reparameterize, right? The problem is, first of all, it's not easy to reparameterize the model. It's not a trivial thing to do. Second of all, even if you reparameterize, right, we see this in hierarchical models where you reparameterize, so decenter, for instance, and then everything's working, and then you add more data, and then it stops working. So
Oh, well-
not even, so different parameterizations will work with different data regimes. So yeah.
Well, yeah,
small.
that is
Yeah.
the fundamental problem, which is that it's the model conditioned on your data. You can't just look at the model and say, this is going to work. This is a one size fits all thing. It's not. It's really about your problem and your data.
Yeah, it makes life for methodology people like me very hard. But at least we get to keep our jobs. Right?
Well, thank you very much for everyone's feedback as well.
Great. So if there are no other questions, I mean, if there are other questions, you can ask them in the chat. I will move on to the next talk, which will be by Martin Modrak. So let me just share that now. I'm going to mute myself as well.
Hello, this is Martin Modrak. I'm going to speak about hidden Markov models and how they relate to Cox proportional hazard models of disease progression. And hopefully that's going to be useful to some of you. So what I mean by disease progression models. So in the very abstract, it's just state models modeling different states of disease. So the simplest is the survival model. We have just two states, alive and death. There's a single transition from alive to death. Other transitions are not allowed. We can go a bit more complex, so we can have like competing risk models where there are two different events and they are disjunct, they can happen both. Or we can go even to more complex models. For example, this is the model I used to model some COVID data set, where people have various requirements of breathing support, and these are all the transitions that are possible in the settings where people can get healthy and get discharged from hospital, or they will need various ways of support, and they unfortunately can also die. So these are terminal states. Or alternatively, similarly complex model from a Williams et al. Study that inspired a lot of what I'm going to talk about, and most of the approaches I talk about were described there, and here they model progression of dementia, and they have low amyloid and high amyloid states, and then each of those states then separately has like 3 states for various stages of cognitive impairment where a patient can progress, and also like from all of the states there's an arrow to the death state, because those are all people and unfortunately they die. And with these abstract state diagrams, we haven't specified how do we actually fit those types of models. And so the most common way to do this is the Cox proportional hazards model that can handle all of those multi-stage models reasonably well. It's a great tool. So there's also the accelerated failure time model, which I'm not very familiar with this, but just wanted to list for completeness. And then our Markov models and hidden Markov models, which are the main focus of today. And so like if Cox proportional hazard is used so widely, so what it cannot do, why would we not use it? And one of the reasons is that Cox proportional hazard model makes it hard to impose some sort of structure in our states. And what do I mean by structure? So for example, in the COVID data set, we want to say that the probability of moving to the ventilated state, given that the patient requires supplementary oxygen, is larger than the probability of moving there for a patient that doesn't need any breathing support. AA here stays for ambient air, is breathing fine. So that's the kind of structure we want to impose. And obviously no structure means our model can be so rich because we can share information. If we have structure, we can share information between various transitions and states, and we have no structure, we need more data to estimate the same number of transitions. And if some transitions are super rare in the data, we cannot really learn much useful about them with the Cox model. Also, Cox can easily handle missing or partially observed states, and maybe there's some way to do this. I'm not an expert in Cox models, so please correct me in the questions time if there's some obvious way I'm missing to do this. But this seems hard to do, especially in the context of, for example, the dementia progression data set. So since we observe patients only one day with the doctor in this data set, so we cannot assume that they change state at the time they are measured. So the first time they are, for example, diagnosed with dementia, we cannot say this happened at the doctor's visit. It had to happen sometime before, we just don't know when. And so what are the Markov models that propose as the alternative? So they have the Markov property, that is the probability of being in a specific state at time t depends only on the state at the previous time point. And this can be somewhat, so in the very basic formulation, we need a distribution of initial states, and we need a transition matrix that describes how the states evolve. One of the things this means is that the baseline risk is assumed constant. So in the simple formulation, and this implies a geometric distribution of sojourn times in a single state. So this can be somewhat alleviated. There are ways to make it less so it's not a hard constraint, it's just like the simplest formulation has its property. Also, it turns out that the models fit pretty well even when this is violated, and the inferences seem to be pretty good in some of these simulation studies I've read, even if this is not true. And it's very easy to make the transition matrix differ in time between patients, so there's no need for the transition matrix to be constant across all states. It just needs to depend only on the previous state and potentially some covariates that are the same, that are not estimated. And we then move to hidden Markov models when we don't observe the states directly. And here we can actually use an arbitrary distribution. In the COVID data set, what we did is we assume there's a small chance that the patient is in some sort of neighboring state. So maybe the patient is in the receives supplementary oxygen, but maybe they would be okay without oxygen. And they just have like oxygen for abundance of caution. So there's some possibility that the patient is like slightly misreported beyond their like actual health state. But for example, in the dementia progression data set, Williams et al. Have like 5 variables that are a function of the state and that can be observed in different ways based on the state. So it's like there's some additional parameters that is like mean amyloid burden in the high amyloid state, mean amyloid burden in the low amyloid state. And these kind of parameters additional that modify the likelihood. And when we have that, there's this thing called forward algorithm that lets us very efficiently compute the exact likelihood of all of the full sequence of observations given the parameters. And it behaves very nicely. It's a nice piece of software engineering. It's not very difficult also to implement. And once again, there's no problem with varying transition matrices. There's also no problem with missing observations on the outcomes. There's like really no problem with that. We just like don't use the likelihood for the steps. We have missing observations and it works. What we cannot have is we cannot have missing predictors. So we need to be able to construct the transition matrices at all time points, but we don't necessarily need to observe the outcomes at all those time points. So how do we construct varying transition matrices? So the simplest approach is just to treat this as a categorical regression. So for each state, we model the probability distribution of moving to the other states as a categorical regression. But it doesn't let us impose a lot of structure, so that's not super useful. We can impose some structure by treating this as ordinal regression. So you may have noted that in the COVID dataset, the states are ordered from discharge to ambient air, oxygen, ventilated, death. So we can treat the distribution of transition probabilities as an ordinal regression, which enforces a lot of structure in the model and implies these kind of things like that patients that are on ambient air are less likely to progress to ventilated than patients that already receive oxygen support. So that's pretty neat. And even more generally, we can use rate metrics, which is what I'm going to talk mostly for the rest of the talk. That's even more flexible. So the question here is like, why do we observe transitions like from ambient air to ventilated without passing through the oxygen state in the model? And one possible answer is actually that's an artifact of the data collection. So there were 2 transitions. So the patient first deteriorated to the state that they needed oxygen, and then deteriorated further to needed ventilation, but both of those happened in a single day. So what we can think of is in this full data set, this transition is fake. It's not a real transition. It's just a compound of this transition and this transition here. Similarly, we can think that this transition is just the patient moving first to oxygen, then to ventilated, then to that, and this all happening in a single day. So it's a bit more complex to think about the oxygen to death transition. So we keep that in the model because some patients will never get ventilated because they're too frail to be ventilated. But we can remove some of the arrows and treat those other transitions as implied in continuous time by the structure of the model. So we remove those arrows and then we think, okay, so we're in continuous time. So how do we describe transitions in continuous time? And one way is to treat it as an ordinary differential equation. So here we'll have that for a single state, the time derivative of being in the ambient air state increases with the rate of transition from the oxygen state to the ambient air state and the probability of being in the oxygen state. And it decreases for each outgoing transition. So it decreases by the transition from ambient air to oxygen and from transition from ambient air to discharge, all multiplied by the amount of the probability of initially being in the ambient air state. And we can make this slightly more general. So for each two states, we have a rate of transition between those states. And the diagonal of this matrix is just the negative sum of the outgoing rates. All the rates are positive, and R is sparse, so there could be a lot of zeros. And then we can treat this as an ODE, as a differential equation. The time derivative of the probabilities is just the probabilities multiplied by the rate matrix. And if we get an additional condition, this has an analytical solution. The exponential here is matrix exponential, so it's matrix exponential, but we can explicitly solve for the probability at time t with matrix exponentials. So given the solution from before, we can use this to construct a transition matrix, so the matrix at time t plus 1. So when we exponentiate the rate matrix, we get that the probability vector at time t plus 1 is this transition matrix times the probability vector at time t. And A will not be sparse even if R is sparse. And we can then put predictors on the logarithm of those rates. We keep discrete terms for predictors because we need if the rates are not constant for over the time unit, then the ODE can be solved. And one downside of this is that the matrix exponential is very expensive, but it's somewhat elegant. So as long as we don't need too many different matrices, it's not a big problem. But with a lot of matrices, it can take a lot of time. And this lets us to build more complex models with the same data because we've imposed some additional structure. So for example, here we have 18 possible transitions. So because the patients can transition from unimpaired directly, for example, to high amyloid dementia, but we believe this has to happen by either going to high amyloid unimpaired or going this way here. And so we only need 13 rates describe 18 possible transitions. For the COVID data set, we also consider it a more complex model where we have 2 hidden states for each observed state. So we have improving and worsening variants of the states, and this relaxes the need for geometric time in a given state because now the patient is allowed to move from ventilated improving to a ventilated worsening and back, so the distribution becomes more complex and more flexible. But we have more states, and now there are 42 possible transitions, but we once again only need 13 rates to describe all of those 42 possible transitions. Now, like the final question, like, is it useful to model data this way? Well, I hope so. I'm not completely certain. I think it's interesting. That's why I'm sharing it. But for example, here with the COVID data set, there's a multiverse analysis, and you see that here those are the results from the hidden Markov, from variants of the hidden Markov models, and they are not so different than the inferences from the variants of Cox models used here. So it's not a big difference, but we were kind of able to use a bit more predictors, use a bit more predictors, and that's probably useful. And we have like narrower intervals despite using more predictors. So there's some sense in which we're probably using our data a bit more efficiently with the hidden Markov model. What happens, we bring in additional assumptions about the disease progression. This lets us build richer models that make better use of the data, but then we need to obviously justify the assumptions, and it's not completely obvious that all of them are completely justifiable. You may want to use this yourself. So where is the code? I actually have a code that wraps this inside BRMS. So you have BRMS type predictors for the rates. There's some discussion of this at those links. The code works. It's not completely polished and neat, but it works. And I'll be happy if you want to use it. So that would be a push for me to develop it into something more broadly useful, because right now it just sits there on GitHub. So if you have a use case, please let me know. I'll be super happy to collaborate with you on applying the model to your use case. So with that, thank you, and I'll be happy to answer questions.
Right, that was very nice. Thanks, Martin. I forgot to say in the beginning that Martin is a researcher at the Czech Academy of Sciences. I kind of hoped he was going to say that in his video, but he was too busy describing the actual method. So we have a few questions here. Have you seen them, Martin? So you can answer them.
Yeah, so the first question is like whether the transition from ambient air to ventilated is actually biologically or medically going to the oxygen state. And I admit, I don't know, like that was a modeling assumption we made. And I don't have, like it didn't seem ridiculous. And I ran it by like my clinical colleagues, but yeah, it's just an assumption. And I wouldn't like make any strong statements based on this. It's an assumption that seems reasonable. But it's by no way like a fact. I would like, it's not a medical fact. Also, like one thing I forgot to mention in the video, like there are some results on hydroxychloroquine, and we don't trust those results. And it's just an example. And I know this topic is heated, so I just wanted to make one thing I forgot to say in the video is that those results, because the data has problems, so all of those results are not super trustworthy for hydroxychloroquine. So that's one thing I want to just make sure. So yeah, so that was one question from Parksheet. So yeah, I don't know. I hope it's a good model of the data, but it doesn't have to be. Yeah. So the second question from Andre is, yeah, that in HMM the transition times are exponentially distributed, or in the continuous time, or geometrically distributed in discrete time, and so you are right that this is a problem. And so there are a couple things you can do. So in the complex model, where you have like the improving and worsening state, for example, oxygen being improving on oxygen and worsening on oxygen, and then the patient can go back and forth between those two states. And this already induces a more complex distribution of the observed state times. And there are a couple other tricks that you can do. Basically, you can increase the number of states and make it mimic any distribution you want. There are some results on that. I might want to share a link here in the chat later, but there are some results on how to approximate any distribution by adding more states. Alternatively, you can complicate the forward algorithm and make it less efficient. And then once again, you can have any distribution you want for the time spent in an individual state. So there's some efficiency and expressivity trade-off here. So I hope that answers the question. If not, please please ask more.
Right. So Frank also posted something, a link to discrete time ordinal state transition models, which I know nothing about. So as a biostatistician, it's a bit weird, a bit shameful to admit that I know nothing about survival models. That is, so I don't know how these approaches kind of compete, but if it's simpler, then I guess it's better, right? So if you can represent the same sort of information with a simpler model, it's always good. I had a question, unless Frank wants to comment on the link he posted.
I would just say simpler would be better, but there is that complication about irregular measurement times or gaps between times that are unpredicted, where the likelihood function gets to be combinatoric if you want to handle the missing data exactly correctly.
Right. How often does that happen, though?
It happens just often enough.
Well, of course it does. Yeah. I should have known better. Go on.
I hope that the model, I think, as far as I understand, I think Frank's model can be seen as like a specific case of the Markov model. I think it could be understood in this way. So you have but I'm not like 100% sure, but I think it could be understood as a special case here that works there with fully observed data. And then I think in this case it simplifies the likelihood calculation, but I think you can compute the full Markov model and then treat like gaps and everything smoothly. That's my understanding. I hope I understand Frank's model correctly.
It's not exactly that, because it doesn't assume exponential distributions for times anywhere.
Right.
So that part can be more general, but the gaps are less elegant than what you have with your continuous time model.
Right. A question I had was how expensive in terms of profiling, right? So how expensive are the matrix exponentials in your calculations?
Like they're dominated computation time. So as long as you have just like a couple, it's not a problem. So one of the biggest models we've read was this. Then included like age of each patient. So you basically had each patient had like one transition matrix before treatment was initiated, then another one after the treatment was initiated. So because like the ages differed, there were very few patients that had the same age. And like for this model, like doing the prediction, so like taking the samples and computing the exponentials without the gradient, without anything, took like 2 hours or something. So yeah, so they're super expensive. And they're definitely like the number of matrices dominates the computational time. And I had to do like if you don't like keep track of how many matrices you need and reuse the matrices, the model just doesn't finish in any reasonable time because the exponentials are just so expensive. But in a lot of cases, you actually just have like 5 or 10 different matrices in the whole data set, and then like it's pretty okay.
Right. Yeah. So in phylogenetics, we also have the problem, right? So all of our likelihoods are matrix exponentials. And in certain cases, there are discretization techniques that you can use called uniformization. We would have to kind of sit down and think a bit more about what are the conditions on your matrices so that this would work. But funnily enough, it kind of involves discretizing. So you went to continuous space, continuous time for your modeling, and then you got a bunch of ODEs, but then to compute this efficiently, assuming it can be done, you would then rediscretize in this uniformization type algorithm. But it's something to look at, because if this is the bottleneck and you can do it well with uniformization, could be a game changer, right, in terms of applicability.
Yeah, maybe well, actually, maybe that was not clear. I actually don't do anything in continuous time. The continuous time is here just to motivate the matrix exponential.
Right.
But then it's just like all of this is to motivate just like a specific way to build the discrete time transition matrix. So that's really what's happening in work. So it's just like another way to construct a transition matrix in discrete time by going to continuous time and looking what would you expect here. So,
right.
I'm
That makes sense. Yeah. Right. So we'll now move on to the last presentation. That will be by Alejandro Nieto Ramos. He's a PhD student at the Rochester Institute of Technology. I'll now mute myself and project his presentation. Just give me a sec.
Hi, my name is Alejandro Nieto. I'm a PhD student in mathematical modeling at the Rochester Institute of Technology. I will talk about a Bayesian approach to representing variability in space in cardiac action potential properties. This is some work that I did at Oak Ridge National Lab last summer. My advisor there was Christopher Crable, but like this is a part of my thesis proposal. My advisor is Elizabeth Cherry from Georgia Tech and Flavio Fenton, also from Georgia Tech. So let's start. So when we want to think about cardiac variability in cardiac electrophysiology, we notice that myocytes or cardiac cells, they show variability in the shape and duration of their action potentials. And this happens among individuals, but also in space and time within a single individual. At the bottom, in red, you can see action potentials. And the problem of finding parameters for a cardiac action potential model to reproduce specific dynamics can be difficult because there's a nonlinear dependence on pacing period and also because of the parameters involved. So you can think of the pacing period as when the heart is at rest and with the heart is like more excited when, for instance, when we are running. So at the bottom, you can see two images of a rabbit heart. They are called optical mapping. And in circle, in the white circle, you see two different locations. And as you can see, the action potentials are not the same for the two locations. The color in gray represents a function of the action potentials. As a matter of fact, it's action potential duration. I'll talk about that a little bit later. So these hearts were paced at 280 milliseconds. So the objectives of this talk are the following. We are going to do Bayesian inference on the Mitchell-Shaffer cardiac action potential model, feed it to synthetic action potential data represented on a grid. We'll do that by defining a hierarchical model on the parameters of Mitchell-Shaffer and simulate spatially correlated values on a 2D domain for one of the parameters using a hierarchical but non-spatial structure for the rest of the parameters. For the parameter represented in space, we are going to use a latent Gaussian process as a prior, and we are going to do inference in a subset of sample locations used as training points and predict values on the non-sample locations using the no U-turn sampler and Stan for posterior inference. Let's see some cardiac action potential terminology to make this talk a little bit clearer. Cardiomyocytes are one kind of excitable cells. And when they receive a sufficiently strong stimulus, the membrane potential changes. And that causes ions to move across the membrane. At the bottom left, you can see a graph that represents the interior and exterior of a cell. And in pink, you can see the proteins called ion channels. They are like gates because they open and close. And when that happens, ions cross the membrane. So when the membrane potential increases above a threshold, we get an action potential. You can see an action potential profile of a zebrafish that is paced at 400 milliseconds at the bottom right. Well, now let's see why this problem is a little bit difficult. The reason is that these are experiments. These represent experiments. So again, this is data from zebrafish. So the cardiac tissue is outside of the body, and it's stimulated by using electrodes. At the bottom, you can see the period of cycle length that is used to stimulate the cardiac tissue. So for instance, at 400 milliseconds, we see this series of action potentials. After reaching steady state, you can see these two red lines that represent action potential duration. That is the time that it takes for a cell to depolarize and repolarize. So after reaching steady state, these two lines are the same. So the action potential is the same for two consecutive action potentials. But if you start pacing more rapidly, you see this phenomenon of a bifurcation that happens at approximately at 300 milliseconds. So for the example here at 275 milliseconds, after reaching steady state, you see that the action potential duration changes between long and short. These are called alternants, and they are important to study because they serve as precursors of cardiac arrhythmias. So when we are going to feed action potentials, we do it not only using one cycle length, but more. Otherwise, it could be like feeding a line with just one point. For this work, we are using 3 cycle lengths that are close to the bifurcation point, 2 causing alternants and 1 not causing alternants. So the methods are the following. This is the Mitchell-Shaffer model. It's a system of differential equations. It's very simple. It's a phenomenological model that has 2 variables, U as the membrane potential, and H represents a gating variable, like the pink cells that pink proteins that I showed you before. The parameters are 5, they are in blue. At the bottom, you can see the solution for this system for a certain set of parameters. In blue, you see the membrane U, and in red, you can see the gating variable, H. So why is that we are using a Bayesian approach? Well, the reason is that we know that variability can be completely described if we know the probability distribution of the variables of interest. In this case, the parameters of the model. Bayesian methods enable fitting a probability model to a set of data. This set of data could be experimental or simulated, and the result is summarized by a probability distributions on the parameters of the model. Basically, the distributions give us the following information. They give us a description of randomness in the observed data, of course. They also facilitate making predictions of unobserved variables, and they provide information about the values that the model parameters can take. Sometimes researchers don't know that, but of course, if they do, they can use that information to put it into the prior. Specifically, we are going to use Monte Carlo methods. These methods can be used to sample from the probability distributions of the parameters when experimental data is given. Traditional Monte Carlo methods or MCMC methods have the disadvantage that they don't explore the parameter space in an efficient manner. So if the dimension of the parameter space increases, they become computationally unfeasible. So that's why we are using Hamiltonian Monte Carlo. It has the advantage of using gradient information from the target distribution, and it explores the parameter space in an efficient manner, even if the dimension of the parameter space is large. The statistical model is basically this. We have a differential equation x prime, and we are going to model the response, meaning the voltage y, as the solution of one of the states of the differential equation plus some random noise that we consider is normally distributed. And something basic in Bayesian statistics is that for the work that we are doing here, we don't know the values of the parameters. They are unknown but fixed. So we consider them as a random variable. And then we use Bayes' rule that says that the posterior, the probability of the parameters given the data, is proportional to the likelihood times the prior. And we are going to sample from the posterior or target distribution. So now here is the structure of the hierarchical model that we are using. So here the individuals are each of the pixels in the image, on the image. But also, so we are going to use priors individually for the parameters individually, but outside of the box, we are going to use also priors for the whole set of locations as a population. So we are doing inference at 2 levels. The structure inside is for the parameters from tau out to the U gate. We use priors that are not spatial, but for the parameter tau in, the structure is different because we are using a Gaussian model. All the circles in white represent random variables. Sigma here is the parameter of the error or the noise, and y are the data that we are giving. That's not a random variable. That's why it's in gray. So basically, this is a structure. And so now, what is that we did? So we created a grid of correlated values. So we just created random values that we know one of the parameters can take. We apply a Gaussian filter because we need the values to be correlated, because this actually happens in the heart. Cells are, electricity is transferred from one cell to the next, so there's correlation. And what we did is at the black points, we gave cardiac action potentials, and we are going to predict the values that are not on those points. And like I told you before, we use 3 cycle lengths. So the results are the following. On the left, you can see the actual values that we used. And in the center, we can see the values that we predicted. So as you can see, the grids look very much alike. And if we plot the true values against the predicted values, we see that they are highly correlated. The bars represent the posterior standard deviation of the posterior mean estimates. We use, like I showed you before, 7 training points, and we predicted 18. And we also calculated the Pearson correlated coefficient that was very close to 1. So that also says that the correlation is high, not just seeing it graphically. So here is an example of what we are doing for prediction. So in the star is a value that we didn't have before. With the values of the parameters obtained at that location, we get these 3 populations of action potentials for the cycle lengths that we use. And well, don't think that you can plug any set of parameters in the model and you get action potentials. So this means that the values that we got are good values because we are actually getting action potentials that make sense, because otherwise we could get log, or we could get that the action potentials oscillates. That's not the case here. So as conclusions and future work, we implemented a novel approach to model variability on the Mitchell-Shaffer model, obtaining promising preliminary results. And we were able to model variability of action potential profiles in space using synthetic data and predicted values on the rest of the positions that were not used to do inference. As future work, we plan to do the same kind of inference or on larger grids, also using a different number of training points. As a matter of fact, I have been doing that. But well, right now I'm working on something else. But I have worked with grids of 10 by 10 up to 30 by 30. We also plan to consider variability in space for the rest of the parameters of Mitchell-Shaffer. And we also would like to use a more complex cardiac action potential model like Fenton Karma that has 13 parameters. But well, basically, we are doing all this because we want to apply the technique on experimental data from optical mapping. So thank you very much. If you have any questions, you can ask me now.
Right, nice. So that was Alejandro's talk. I've already posted a question on the chat, which I can and will repeat, but please also feel free to well, ask questions. So the question I had was, Alejandro, whether you experienced some sampling problems such as divergence or poor efficiency, you know, what's the computational behavior of this model with the data you have?
Well, yeah, it has, I'm not unmuted. I have seen that for certain set of parameters, there are a lot of divergences. So I suppose that for certain set of parameters like the dynamics don't help, and I have used different priors. I have used folded normals, I have used gammas, and I still notice that problem. That especially happens with where I have taken the models for synthetic data from papers. For instance, from Fenton Karma, if I use the data that comes from the paper, I have never been able to not get divergences. And you know, I noticed that I had to run the script that I gave you because I wanted to see that everything was working well. And I noticed that there are, I think, more divergences than when I did it a long time ago. So yeah, I have noticed that.
Right. Yeah. So that was my first thought when I saw that hierarchical structure, that you might find these problems. And the problem is that it might be nothing, or it might be, you know, some weird degeneracy in the model that makes it hard to actually infer what you want to infer from it. And the next thing I want to say was not really a question, is that, and it's a more general comment, I think, about the applications we've seen today, is that you really have a lot of room for some interesting prior and posterior predictives, right? So in your case, you have domain expertise that you know what the potential curve should look like. And it's interesting that you can then check whether your model is doing something reasonable with that. So you can look at the posterior predictive, and I would advise you to also do the same with the prior predictive, so you understand what your priors imply on the space of the things you care about, which is, you know, something we as a community have been slowly kind of waking up to, even though some people have been telling us about this for 40 years. We're now, you know, at the point where our models are so efficient, our samplers are so good, that we can do loads of simulations to try and understand what our priors on certain parameters actually imply on the whole model, not only quantitatively, but also qualitatively. And I think your application is a good example of that.
Well, you know, a way of seeing that the prediction is good is that, you know, that for the training points, I could have plotted the population of action potentials that I gave. So they are in fact centered like between the population. Right. And well, so I don't know if you were saying that I know the priors, but not necessarily, I mean, I know the support that the priors can take, but maybe no, not necessarily. What I did to get those values was that previously I was working with ABC, SMC. So I saw that it was very useful for me to get good values for the support of the priors. So my idea would be that since ABC, SMC is so fast, I could like use a combination of the two methods. And I could get like values for the priors from ABC and use them for HMC.
Right. Yeah, I mean, I think Ethan's comment gets at this question as well. It's, you know, he says I think it's important not to overstate the performance of HMC compared to traditional MCMC. I think there's truth to that. Well, we're all here because we love Stan, right? I think. You know, Stan has been really good. Well, I say Stan interchangeably with HMC because the implementation in Stan is a particular implementation that makes loads of things that were a pain in the past makes it easier. So picking the step size and picking the size of the trajectories is a pain, right? If you want to do HMC by hand, it's an inferno, right? So Stan makes it easy to have something that works quite well for a majority of models, but certainly there are problems that we can't solve with Stan, even when we are clever about marginalizations and other things. I recently worked on something close to what I posted here in response to Nick's talk, where the posterior is so constrained that basically Stan could possibly move. Right, I got it to just work in the end, but you know, it was very constrained, and the differential structure was a nightmare. So there's certainly room for many different algorithms, right? So I'm working right now with something with a student, Lucas, who's here, and we might couple Stan with other samplers and do a kind of Gibbs thing where we switch between the two. So we get the samplers to do, you know, what they are good at, right? And we get a nice sampler, a nice meta sampler in the end. Right. Does anyone have any more questions? Questions or comments on Alejandro's presentation? Right. So we now will have a few comments from Frank Harrell. I'm very, very happy to have him here. And I don't think I have been very appreciative of your presence here. So I would like to take this opportunity to really thank all of the speakers for taking the time to prepare their presentations in advance and be here. And I would like especially to thank Frank for his time and for agreeing to be a discussant in our humble event. So Frank, the floor is yours.
And Luis, thank you very much for inviting me. The talks really covered a wonderful spectrum because we had population disease mapping, we had biology, biochemistry, physiology, and clinical studies. So just wonderful coverage. And I'm not going to tell you how wonderful each talk was because we're taught that if you're writing repetitive code, that's a bad idea and you need to factor that out into a function. So my function is to state that the talks were just uniformly excellent. So I want to congratulate all the speakers on giving excellent talks based on excellent research. So I'm going to start with a brief discussion of Mitzi Morris's talk where she went over the Byom2 model dealing with spatial disease mapping and including possible non-spatial components and complexities such as disconnected islands. Mitzi found that Stan was expressive enough to be able to describe these interesting configurations. And so she used a shapefile, and then she had a different notation for disconnected components. She used at the heart of it a Poisson likelihood, and the Stan code was very elegant. It does get messier for disconnected regions, and Mitzi emphasized the importance of vectorization. It would be good to hear more about choice of priors in her model. and she voiced a need for ability to handle ragged arrays. And there was also discussion of the need for vectorized CDFs with some possible things coming in the future in that. Next, Ethan Alt gave a talk about normalized power priors. And a sub point there is there needs to be a certain level of pre-specification, because this might be involving comparing treatments where you're coming before a regulatory body such as European Medicines Agency or FDA. There are some difficulties with hyperpriors related to that, but the method that Ethan talked about had a particular challenge of an unknown normalizing constant. So he used an MCMC approach where you have samples from the prior, and also did an importance sampling approach, which was faster, but it doesn't work when there's a lot of parameters or there's a dispersion of parameters. So I think what I would like to see from Ethan is a little more motivation for the use of power priors in general, because they create complexities, and it has to be worth the trouble to have that level of complexities. And in my time at FDA, we discussed this a lot, and there was sort of a little bit of a movement away from these kind of priors, not in any big way, just in a subtle way, more in favor of things like mixtures of priors, because when you have a mixture of priors, the mixing proportion has a direct interpretation, has an exact interpretation of the probability of applicability of the historical data. So, and the mixing is something you would have to elicit and completely pre-specify. But other than that, mixtures of priors have some appeal because of their direct interpretation for applicability. Teddy Groves went into chemical reactions, enzyme catalyzed reactions, and dealt with kinetic parameters, which are available in some online databases, but it takes intensive literature review to really come up with the kinetic parameters you need for a specific application. So the goal Teddy enunciated was to summarize the kinetic information with a statistical model. And the team and he put in a lot of thought into picking the measurement model. They chose a ANOVA-like model with interactions. The interactions were two-way interactions each. And then they used an approximate cross-validation approach to look at the performance of the model. And uncertainty in the parameters was quantified with quartiles of the posterior distribution. Just a couple of suggestions. One that would be easy to deal with is to look at the distribution of the absolute errors in trying to mimic the kinetic parameters for what we have from known experiments. So the absolute error could be quantified in some sort of posterior distribution. And then I gather at some point logs are taken, and you just need to make sure that before you have an additive model that log was perfectly correct. So there might be just a little bit of work done about transformation uncertainty, just to see if there's any uncertainty in taking logs, if I understood where logs were taken. Nicholas Coey also talked about kinetics, and in particular standardized rate laws, had a very nice simulation using the prior versus the prior plus the measurements, and had to deal with boundary conditions and dealt with those very nicely. The inputs to the process involved the kinetic model definition, the measurements themselves, the prior and sampler instructions. And there was attention to reproducible research, which is maybe something the other speakers could add a little bit to the next time they present this. I think it's always important to state how you would make this totally reproducible by others. Nicholas mentioned the challenge of high dimensional correlations, and did mention there's an expensive differential equation system evaluation at every step. That challenge is probably going to remain there. Martin Modrak gave a talk that's really close to my heart because this is the area I've been working in almost full time since COVID started. So he talked about a hidden Markov model as an alternative to survival analysis. And I think that's a perfect way to phrase it, because survival analysis tends to be the first tool people think of. And it's not very natural because, as Martin mentioned, it doesn't handle missing data in the middle of the observation period, and it doesn't handle the disease progression model. It doesn't handle different levels all at once. So with COVID-19, you might have need for oxygen, then progressing to ventilation, then death. And Martin made a great point that you're using structure of the model to handle cross-state information sharing, and also allow you to deal with partial information when there's a gap in the data collection. The hidden part of the hidden Markov model relates to a kind of measurement error in the states themselves, and a forward algorithm was developed, and this really helps you in dealing with missing data. Transformation matrices could be varying. He mentioned what I hoped he had mentioned, that this also handles ordinal situations. You can put this in an ordinal regression framework. We had a little discussion in the chat about the use of rate matrices versus discrete time Markov models, which are much easier to handle, but they don't handle sort of ragged time measurements as well as having a rate model. The thing that Martin did not emphasize enough, he mentioned the power gain from doing all this might not be huge. Well, I've got on the web page that I gave in the chat some example simulations where if you use a multi-state transition model instead of time to recovery with a Cox model, the power change can be tremendous. So in one example, just analyzing time until it took people to get home, time to recovery, and using a Cox proportional hazards model, the power was about 0.25, whereas the transition model had a power of about 0.7 for the same design. So you're not talking about trivial differences in power. In some of these cases could be huge, and it has huge sample size, and getting the study done earlier ramifications. Alejandro Ramos had a paper about physiology, a talk about cardiac action potentials and variations across individual space and time, dealing with the Mitchell-Shaffer cardiac action potential model. Used a latent Gaussian process prior and had to deal with a system of differential equations. Had a nice listing of the benefits of the Bayesian approach in this particular setting, and including Hamiltonian Markov chained Monte Carlo. The inference is at two levels, the first level being a population level. He showed excellent agreement between true and predicted values. The thing that is not quite obvious from that kind of agreement is their second order agreement in terms of correlation patterns. So the correlation of the raw measurements is modeled, as I understand it, as random effects. And there might be a serial correlation model that would fit a little bit better. So whenever there's serial data, I really recommend people do the variogram or semi-variogram, which will show the correlation pattern versus the time gap, to see if it's an equal correlation pattern like a compound symmetric, which is what random effects use, or whether you have a exponential decline in the correlations or any other pattern, that if you modeled a little bit better, the analysis might be a little bit more powerful. So I think I'll stop there and just voice appreciation to all the speakers. I learned a lot, and it also points out how much more I still have to learn. So thanks.
Great. Thanks, Frank, for a great summary. I think the people will probably want to respond to some of Frank's comments, so feel free to do so. We're now at the official kind of end to our formal part of the presentations, as formal as it was, and now we have time to chat until around about 2, but we can go on a bit longer if you want. So yeah, let's chat. I had a few questions in mind, but I'm organizing them. I did post something about the variogram though in the chat, so it's a bit of an esoteric thing that I learned about in geostatistics, but it's as Frank says, it's a great tool to try and understand the data first, right?
Yeah, it's really
the structure.
It's the QQ plot of time. It's the QQ plot of serial correlation.
Right.
I have one thing I could bring up. I put kind of an obtuse reference to this in the chat, but when you have a discrete time transition model, like a Markov model, and your time measurements are fairly regular, so you don't have to deal with a really complex likelihood. The likelihood using the conditional independence assumption in the Markov model is so amazing. And Stan converges so blazing fast. It's really something to watch. And the nice thing about that setup is you don't get into anything remotely looking like a matrix exponential. The closest thing you get into is a recursive matrix multiplication, and that's only after the sampling is done, because that's only needed to get unconditional state occupancy probabilities. Right. So if you want to move from transition probabilities to the unconditional state occupancy probabilities, you do this matrix multiplication that keeps going over time, and is something you do on all your posterior draws when the sampling is totally finished. So it's a beautiful thing computationally. Just kind of a random thought.
Nice. Yeah. So one thing I didn't catch is how fundamental is the problem with the ragged structure? How unsurmountable is it? Is it completely unsurmountable? Like, do you think there's room for some representation that will keep the computational efficiency of the simpler model, but also deal with different sampling patterns?
The website I listed, I have some simulations of the performance of an approximate method. Cool. So the approximate method works best when your gap times are random. If they have a beautiful distribution, and so if the gap times are not collinear with absolute time, right, then you can sort of separate the effect of the gaps from the effect of absolute time, and then you don't have an identifiability problem in that case. So you can have the gap as a covariate in your transitions, right, and get an approximate decent answer, but the exact answer will come from the combinatoric likelihood that takes into account exactly where the gaps are. And that is where you get into things as expensive as the matrix exponential.
Right. So if you were to think of a regular grid, right, where the measurements at certain points weren't measured, then the model you're describing would be missing at random, right? So you measured the regular grid, and then
if the gap is at the end and you have the missing at random assumption, then it's perfect. It's trivial. Right. So we're talking about gaps in the middle. So if you measure weekly for 7 days, you measure for every day for 7 days, and day 3 was missing, then when you do the transition, what's probability of transitioning into state I on day 4 given day 3? Well, you don't have day 3. Right. So your conditional probability is not defined. And so you have to calculate the unconditional probability conditioning out day 3. Yes. And if day 3 was the only day that was missing, that's a very simple matrix product. If you had day 3 missing and then day 5 missing, then you get into much more complicated unconditioning on day 3 and day 5. So that gets into more the combinatoric problem. And I would love somebody to solve that, because then the discrete time model would be having all the advantages and none of the disadvantages. Right. Yeah. So I was thinking, that you could supplement the space with the variables that you don't observe. So you're observing at irregular times,
but you're going to pretend that you were actually observing in regular intervals, but you didn't get to observe. So some of those values were missing. Then you could try and model, the missing pattern. But as you say, the problem is that the marginalization is not trivial. It's quite expensive.
You could use multiple imputation. It's just that with a full Bayesian solution, we try to not use multiple imputation when we can get away with it.
Of course, yeah, that makes sense.
But there's a lot of interesting issues there, I think.
Yes.
Yeah, so maybe actually I'm missing something. And there's a kid with me, so I might have some noises in the background. Sorry. My understanding is that when you treat this as a hidden Markov model, then you can compute the transition matrix using the ordinal assumption. So you assume that the transition from state, say oxygen, to all the other states is described as ordinal regression, and that column of the transition matrix can be described as an ordinal regression, or you compute the probabilities as you would probabilities of ordinal regression. And then this seems to me to be very similar to the model that you described. And then you can use the full hidden Markov model approach with just like having missing data. And it seems to me that should just work. I didn't try it, but it should just work and produce something very similar and work very well with gaps in your observations, as long as you don't have gaps in your predictors, so that you can like construct what you believe where the transition matrices at the time, if the transition matrices should be a function of some predictors. So that makes sense. Maybe I'm not super clear.
And are you referring to discrete time model or continuous time model?
Yeah, a discrete time model. So it's just a hidden Markov model, and it's just you just construct the transition matrix as you would build an ordinal model. So you have like an ordinal regression for each of the transitions. Does that make sense? Like I'm not sure.
I think the reason the problem exists is when you have a long gap, you're conditioning on previous state, and this previous state is previous to previous to previous. And so the previous state is out of date and it loses, you have an information loss. So you're just not able to condition on the previous day because that day isn't there. So missing at the end doesn't cause any problem, but missing in the middle does, because you don't know what to condition on anymore. So you have to go back and write down the unconditional distribution of all the days that were measured. And that unconditional distribution, if day 3 is missing and day 5 is missing, is a very complex sum of matrix products.
Right.
So maybe I'm overthinking it. Just my impression is that the forward algorithm gives you exactly that, like in some cases. But maybe I'm missing something. I probably need to read it more thoroughly. Maybe I'm missing something.
Yeah, I don't think the forward algorithm works as simply in discrete time, but I would love for you to show me otherwise. And let me mention that for Alejandro's presentation with modeling the action potentials, I think a Markov model could possibly be working there too. And that one, it has regular data points, so that could be continuous time or discrete time model would both work equally well. And that would handle a very complex correlation pattern.
Yeah, you could work out some approximation, some Markov approximation to the more traditional models, and then sort of show that it works way faster and better.
And when you use an ordinal model, Markov model, the outcome can be continuous. So, an ordinal model handles continuous measurements too.
That's true, because the latent thing is continuous.
Frank, I just wanted to say thanks for the comments, and I do kind of agree with you in the sense that there has to be benefits to deal with the type of complexity of treating a parameter as random if it involves like more estimation. But I guess I will say I do think one of the biggest advantages of the power prior is its interpretability as a discounted likelihood. Like to me, that's a lot simpler than some arbitrary normal distribution in terms of, or mixture normal distributions, if you will. Because even if you ultimately have a small historical data set, there's no guarantee that asymptotic normality in the posterior is going to actually hold well, approximate well. And in such a case, the power prior kind of gives you an automatic way of being able to still utilize the historical data without relying on asymptotic approximations. Yeah.
Just quick reactions to that, Ethan, is that when you think about the power prior as giving you a discounting of previous data, and you can calculate that pretty easily in terms of effective sample size that you're using from the previous data, that's the great part of it. So I really like that. But if you ask in the interpretation of how much weight you're giving previous data, the mixture priors has this exact interpretation, which is the probability that the previous studies are applicable to the current efficacy study. So that probability of applicability is very appealing to regulators.
Yeah, I certainly agree with that. Like the interpretation is quite nice in that regard. But as a comment to both methods, one thing that Matt and I were kind of discussing was that there's this kind of false idea that when you treat a hyperparameter as random, that it automatically gives you, quote, the right result. When in actuality, you're really just accounting for the uncertainty surrounding what that value should be. And if you were just to integrate out that hierarchical parameter to get the induced marginal prior, it would actually give you the same result. And I think that's something that's kind of underappreciated with Bayesian hierarchical models.
Right. Yeah. What I will say is a kind of related comment is that linear mixtures, when we say mixtures, first of all, we usually think of linear mixtures, but there are other ways of, mixing distributions. Linear mixtures are nice, interpretable, and tractable, but they can do weird things, right? So for instance, there's this thing called relative propensity consistency, which basically says, if everyone says that A is more likely than B, then your pool, your mixture should preserve that, right? It should say that A is more likely than B for events A and B. Now, linear mixtures don't necessarily do that. And then if you want to really have that, you're going to have to do log linear mixtures. But log linear mixtures are a pain to use in practice because you have to compute a normalizing constant. You raise the densities to a power. It's weird. So, I guess my comment is you win some, you lose some, right? So you have an interpretable mixture, but then it might do weird things. And in the power prior case, the normalized power prior case, sure, there's a burden of intractability that you have to take on. But then one thing that we can show is that if data sets are incompatible, for any incompatibility, the A naught parameter will go to 0, right? So eventually you will account for the fact that you shouldn't be using that information. So it will discount as well. The other direction is a bit weird. So you can have two exactly equal data sets. And we are in the process of proving that actually the posterior distribution will not concentrate around 1, although its mode will be around 1. So it has some interesting properties in that sense as well. But no one thing is the answer, I think. It really depends on the regulatory question, I suppose. I have no experience, and Frank has a lot of experience with the FDA. So
absolutely.
Yeah, he should be the judge of that, I suppose. Right. Do we have any more comments? I have a question, a general question. I will address it to Frank, but everyone else should feel free to jump in. Those of us, especially those of us with teaching jobs. One thing that this event today made me realize that I should really have realized way longer ago is that the stuff that we teach students is really far away from the stuff that people end up doing in their research careers, right? So I'm teaching statistical inference this semester. You know, the models I treat there had nothing to do with the stuff that we do for reals, you know, in the real world. So, you know, how do we bridge that gap if it can be bridged at all?
I think the best way to bridge the gap is to find out who's teaching the intro stat courses and try to find out what their life expectancy is, so we know when to plan on making a change.
All right. Yeah, I would like really very much to change things.
Intro courses are ruining us. And then you spend 10 years of your career trying to unteach students what they learned in their first courses.
That is true. That's a bit like how I feel as well. You know, I teach things. So right now I'm teaching hypothesis testing, which is both a pain and I don't see the point of, because I use very little.
It's bullshit. It's all bullshit.
Yeah, it's all bullshit, but I can't say that because my job depends on teaching the course well.
Tell them it's bullshit while you're telling them how to do bullshit.
That is possible.
You know
That's a bit of what I do. Yes, Connor.
Oh yeah, that sounds rough. I'm glad I don't have to teach that right now. But you know what? I don't see why you can't teach that. I mean, this morning I was reading Fisher. Scientific inference. What did he say? He critiqued the hell out of hypothesis testing. He was also critiquing Bayes' theorem in like same passage, but he actually had a very good critique of what people are doing today.
Yeah. What I try to do is really make the students aware of the pitfalls, right? Instead of directly criticizing and call it bullshit, which might disincentivize them to study for their finals. What I do is I really try to emphasize where these things can go wrong. But it remains to be seen how effective that is. You know, they're going to be somebody else's students, and they'll probably complain with me that I didn't train them well.
Let me just say this, and then I'm going to shut up and hope other people chime in. The Richard McElreath book, Statistical Rethinking, is truly revolutionary. I mean, to have a first stat book that doesn't use any frequentist ideas in the whole book and shows you how logical statistics can actually be. And just imagine an intro stat book that teaches you how to deal with missing data and measurement error. It's because of the Bayesian framework that he could bring that into an intro first course.
That is true.
This is revolutionary. And until that becomes the first book that people are taught in stat, I think we'll have our work cut out for us.
Yeah.
Well, certainly,
go on.
We certainly recommend that book at every chance we get.
Yeah. So we actually have a few statistics courses, you know, so we offer 2 undergrad courses, applied maths and data science, right? And we have like 3 courses in stats, so statistical inference, statistical modeling, and time series. And there's actually a big disconnect between statistical inference, the course I teach, and statistical modeling, which is the next course they will do, well, next year. In that course, the statistical modeling actually uses Michaelis' book. And I've been trying to work out the politics, so I get to teach that course and relegate the inference one to someone else, because I can't be asked to prove the Ryle-Blackwell theorem once more. But anyway, that is a wonderful, wonderful book. And I've been watching some of Richard Michaelis' talks and lectures on causal inference as well. It's just lovely stuff. I hope we can refurbish, reform our courses so they all kind of look a bit like statistical rethinking.
Yeah, I think a big problem is just that there's such a long history of teaching statistics, and before simulation and all these other computational tasks were even possible, the only thing that we could rely on was asymptotic theory, looking at z-tables and stuff like that. And it seems that despite we have this change and we now have the ability to do such things on the computer, the pedagogy hasn't changed at all. So I think that the real challenge is simply convincing the older people in the departments who operate the undergraduate curriculum to kind of revolutionize it. Because I mean, let's be honest, we all have to sit through intro stats and think it's like really boring. And I hate to admit it, but the fact of the matter is that it is. And statistics really only gets interesting at a very late stage.
Yeah, it's a grind until there. Yes,
well,
completely
there's
agree.
several things I'd like to say, which is that it's not just the senior lecturers, it's also the gatekeepers to publication. So that's really a place that I think everybody here could have some impact on. But also my experience, because I don't teach, I don't have a PhD. I spent 10 years or so, a dozen years, being a support programmer in bioinformatics. So I was seeing all of your students when they got out of class, what they said. And believe me, they didn't get anything out of those classes except a fear of statistics. They were really interested in the phenomenon, in the biology, in the fascinating, amazing experiments that they were doing. The genomics revolution was upon us, and we were finding out super cool stuff every day through fabulous chemistry, amazing lab techniques, really, really, really cool experiments. And statistics only came in when you wanted to put some window dressing on what your theory was and what your gut told you so that you could get it published. You want a report from the field, got a long way to go. Right. But I think the other thing is that this is still an experience everywhere, in that most of the people who are coming through the system aren't going to go on to be researchers or innovators. They're going to be going on to people who are doing useful work, who are applying what they've learned to do standard practices, but to do it well and competently. And there is a lot that's missing in the curriculum to teach people good practice in terms of real world workflow and putting ideas into production. And I would love to, and that is what I hope to be working on a little bit, because my time has been spent bouncing between hanging out in the research world and working in companies. And we really have to address the day-to-day of what people are doing with the ideas we're putting out.
Very well said. Thank you.
Well said.
Yeah, we have a lot of work ahead of us, and for me it's a bit weird because I'm a scientist first and a statistician later, right? I started as a scientist. I'm a microbiologist by training, and then I have to kind of forget about all of the interesting things that I've learned over the years, so we can get to the nitty-gritty of proving some theorems that may or may not be useful, right? So yeah, there's a lot of rethinking to do, really. Michaelis' book title is informative in that sense. There's a lot of rethinking to be done, so we can teach students the stuff that they need to learn about, like bias variance trade-off, in an interesting way that kind of connects with the stuff they're going to see in the real world, and not just as expressions and numbers in a slide. Right. Does anyone have any more comments?
It's lovely to see you all.
Sorry. Somebody said something. Was it Connor? No? Sorry, Frank.
I thought I heard somebody trying to talk.
Yeah, I did as well. I don't know who it was.
Mitzi?
Yes, I have terrible timing. I just don't know how to break into the conversation, but I was just going to say it's lovely to see you all, and putting faces to people's names. Connor and I have been exchanging emails, and it's really, thank you so much, Luis, for organizing this.
Oh, it was really, truly a pleasure. So again, thanks everyone for being here, for being available. This was lovely. We'll have a chat later via email about sharing this materials. But right now I have a very, very upset 3-year-old to deal with. She needs daddy and she needs him now. So thanks, everyone. It was lovely to have you. Bye-bye.
Bye.
Stay
Bye,
safe.
everyone. Organizing.
Stay safe,
Bye.
everybody.
Thanks.